Okay, so hello everyone. So thank you for coming to this presentation and thanks you for the organizers for this nice workshop. So today I'm going to present you some of my recent works about the emergence and the maintenance of modularity in neural networks with ABN and anti-ABN inhibitory STDP. So this is work that I've been done mostly during my PhD with Alessandro Torcini and Matthias Qua in Paris and with Gorka Zamar Lopez and Gustavo Deco in Barcelona. So first, after contextualizing a bit this project and presenting you the specificities of our model, I will move to the two main thematics of this presentation that are the emergence and the recall of modular connectivity and their consolidation and long-term maintenance. So several studies have shown that the brain organization follow a modular structure at both at a functional and structural level, following a segregation of information with the specialization and the brain of brain region and the modalities into modular structures, and their correct integration with the coherent connection and propagation of sensory modalities by hub neurons. However, how precisely these modular structures emerge and maintain over time remain elusive. On the one hand, genetic instruction determine this initial organization and the sensory information pathway. And on the other hand, different factors such as neural activity, sensory experience, or environmental stimuli shape this organization. So as a result, this connectivity appears to be largely induced by various plasticity processes. On the other side, one interesting phenomenon is that during no-task activity of the brain, like sleep or rest, it has been observed asynchronous and irregular dynamics punctuated by partially synchronous and spontaneous activation, that can be compared to spontaneous recall or replay of the learned information that are often associated with memory consolidation. Such mechanisms have been reproduced in several theoretical modeling. However, in such modeling, the synaptic plasticity is not always maintained or applied to all connections, like inhibitory connections, or sometimes they are using a lot of different additional homeostatic or control mechanisms while not avoiding a long-term forgetting of the structure. So at the end, the objective of this presentation first is to find out how inputs can be learned to shape modular structure and memories for simple and realistic synaptic plasticities. Then the objective is to find how these memories can be spontaneously recalled, emphasizing the role of inhibition in this process, and how these recalls can promote consolidation and long-term maintenance of these memories in an asynchronous irregular states. So to do so, throughout this presentation, I will consider a network of n heterogeneous quadratic integrated fire neurons, all connected, where the membrane potential of each neuron is given by the following equation. So without going too much into detail of the equations, the important parameters to keep in mind are first, the external inputs that takes the form of a time-varying external stimulation during learning. Then we have the synaptic inputs that represent the inputs from the other population of neurons. Actually, the specificity of our model is that in that case, we consider three populations of neurons. A population of excitatory neurons following Nebbian learning, a population of inhibitory neurons following again Nebbian learning, and another population of inhibitory neurons following Nebbian learning. The choice of having two populations of inhibitory neurons comes from the fact that in biology, and especially in the cortex, although inhibitory neurons represent about 20% of the neural cells, they are composed of a wide variety of type cells having their own properties and own plasticity functions. So to come back to the synaptic inputs, their dynamics are given by the following equation, where we simply accumulate the incoming spikes modulated by the synaptic weights that is subject to adaptation. This way, according to the nature of the presynaptic neuron and depending on the time difference between the last spikes of the postsynaptic neuron and the presynaptic neuron, we'll define the following plasticity functions. So first, for the excitatory neurons, we'll consider the classical Hebbian STDP asymmetric function from B and Poo that basically potentiate the weights in a causal way. Concerning the first population of inhibitory neuron, it will follow an Hebbian STDP symmetric function that potentiate the weights when the spikes are correlated and depressing them when they are uncorrelated. And for the last population of inhibitory neurons, it will follow an anti-Hebbian STDP symmetric function that, on the contrary, depress when they are correlated and potentiate otherwise. Finally, we also added a small constant term that's supposed to mimic the small and constant forgetting of the information. So now that we are defining the different plasticity functions, we can describe the evolution of the weight with the following equations with our plasticity function for potentiation and depression. Then we have the learning timescale that governs the learning rate of the weights. We also added a small softball function that just allows us to keep the weights between 0 and 1 for when the presenting neuron is excitatory, and between -1 and 0 when it is inhibitory. So now that I hope we have more or less the model in mind, we can move to the experiments. So the protocol considered is quite simple. So where the objective is just to find the condition in terms of plasticity and inhibition to create decoupled modular structure and to obtain a realistic activity at rest with first asynchronous irregular dynamics and spontaneous recalls of the learned information. So to do so, the protocol is divided in 3 phases. First, we have a phase of spontaneous activity, allowing just to visualize the original state of the network. Then we have a training phase where we stimulate 2 different areas, so a group of excitatory and inhibitory neurons, alternatively and randomly with a constant external positive input. And we have a final phase of spontaneous activity where we just observe the dynamics of the network during the rest state, so without any external stimulation, but still under the action of the continuous adaptation. So let's start with the first condition, considering only excitatory neurons in a network. So what we rapidly observe is an explosion of the network activity with a runaway excitation, leading to a fully coupled matrix without structured module. So to solve this problem, we directly introduce inhibition in networks with the population subject to anti-Hebbian plasticity. So in the initial conditions that are a completely random matrix, we observe an asynchronous irregular states. Then during the learning, we logically observe an increase in the firing rate of the neuron receiving an input, while the inhibition shut down the activity of the neuron of the opposite cluster. After this learning, this leads to the following connectivity with 2 modular structures among excitatory neurons and feed-forward inhibition, where each clusters have a population of inhibitory neurons targeting the neurons of the other populations. However, at rest, we still observe this explosion of the network activity where only one cluster firing, shutting down the activity of the other one. So this phenomena can basically be explained by the winner-takes-all configuration that we obtained, since when one population activates, it directly stops the activity of the other population. So to solve this problem, we now consider Hebbian plasticity for the inhibitory population instead of the anti-Hebbian one. So the initial conditions are the same as before. Concerning the learning phase, we still observe an increase in the firing rate of the stimulated population. However, in that case, the firing of the other population is not totally interrupted. Anyway, after this learning, we still have the formation of the modular structure between excitatory neurons, but also among the inhibitory neurons, with the formation of a feedback loop between the excitatory and inhibitory population of each cluster. Now, this structure leads to the emergence of some synchronized patterns of the neurons of the same clusters. However, this reactivation can appear at the same time in the sense that, as we can see in this schema, the clusters, the population are independent. So they are not interacting between each other. So nothing avoid them to reactivate at the same moment. So as a last scenario, we now consider the case where we have the three population, so the excitatory population and the two inhibitory population with anti-Hebbian and Hebbian plasticity. So the initial and learning phase is the same than before. And after that, we logically obtain a structure that is a mix of the two previous one with feed-forward inhibition. So the anti-Hebbian neurons target all the neurons of the other population. And we have the feedback inhibition that target the neurons of the same clusters. Concerning the activity at rest, now we have synchronized pattern of the neuron of the same clusters, but we don't have any synchronization between clusters. So as we wanted, we have some asynchronous irregular dynamics punctuated by the spontaneous recalls of the learning time. So far so good, we have our recalls. But now the question that we probably ask is, what is the role of these recalls and how to consolidate and maintain this structure in the long term? So to answer this question, we started from more or less the same connectivity as before with the two modular structures, but with slightly lower weights, around 0.7, and with random intermodule connections. Then we just let the network evolving spontaneously over a long period of time of 4,000 seconds. So during this spontaneous activity, we observe the recalls that are basically characterized by an increase in the firing rate of the neuron of the same clusters and an increase in the synchrony. And quite interestingly, these recalls are also correlated with an increase in the mean change rate of weights of these clusters, meaning that when we have a reactivation, when we have a recall, the connectivity, the weights of the same clusters increase. So we have clear consolidation of the connectivity when we have these recalls. On the contrary, during asynchronous irregular states or periods that are characterized by a low firing rate and a low degree of synchronization, we have a slow decrease in the module connectivity. Nevertheless, this forgetting is largely compensated by the recalls, as we can see in the weight matrix at time 4000, where the structure is maintained but also reconsolidated over time. So at the end, we end up with exactly with the same structure that we have seen in the previous experiments. Actually, we can even go a bit more further by considering the same matrix, but without the connection involving immediate neurons. So at the end, we just keep the structural module between excitatory neurons. And quite interestingly, we still observe the spontaneous recalls of the learned items. The effect of that is if we wait enough time, we see that the connection involving inhibitory neurons reappear and reconstruct themselves in the long terms. Also, this experiment allows to show that the structure remains stable after a long time, and actually after more than 24 hours of spontaneous activity. So, in conclusion, the take-home messages of this presentation are: first, we have seen that the temporal attention of the external stimuli allows to form structural clusters that are associated with memory items, where, in the one hand, feedback inhibition that are provided by the Hebbian STDP regulates the network activity by establishing an excitatory-inhibitory balance that prevents runaway excitation. Also, this same Hebbian inhibitory neurons allows to promote synchronization dynamic within the structural modules. And on the other hand, feed-forward inhibition that are provided by the anti-Hebbian STDP promote input selectivity and lead to desynchronization between neurons in different clusters. So at the end, these two groups of inhibitory neurons are really necessary to guarantee the emergence of the modular structures and their spontaneous recall at rest. Spontaneous recalls that induce a strengthening of the synaptic connectivity, enabling an autonomous memory consolidation process, preventing the long-term forgetting despite continuous adaptation and dynamics. So I thank you for your attention, and I will be happy to answer your question.
Thank you very much. and the questions there are for you. And the first one comes from Thomas Nowotny. And he asks, why did you use QIF neurons? And was the usual minus V term in the QIF equation missing on purpose?
Why QIF? Basically, before this study, we started to investigate this problem of consolidation of memories with theta neurons. I don't know if you know this model. Basically, the quadratic integrated amplifier neurons is directly related to this one. So this is just the reason why we moved to quadratic integrated amplifiers. However, we have collaboration with some other person who's tried to reproduce this phenomena with leaky integrated amplifiers neurons, and it seems to work as well. So the model is not so important. And sorry, what was the second question?
The second question is, there's no minus V term in your equation, which would make this fire all the time without input.
Minus V, if I'm not wrong, the minus V is only present in the leaky integrated amplifier. The quadratic integrated amplifier, we just have the square term.
I believe there you're wrong, because it also has a leak term, right? So this is just implementing spike, but that's I think that's what Thomas is exactly asking. But this is what you simulated, because your spike, your activity looked like as if there's some leak at least, or enough inhibition for that matter. That's also possible.
Yeah, actually the thing is, the stability parameter that is there is around 0. So there is some neurons that are basically near bifurcation. So you can consider this term as a leak for some neurons.
Okay. Good. So then the next question is by SPK. They were asking, is there any mechanism in your plasticity rule that prevents the weights from saturating?
Yes, in the sense that said there, we have a soft bond function. So the weights are, let's say, between 0 and 1. So yes, cannot saturate.
They do saturate, so there's no mechanism that prevents
No, they cannot saturate because of these terms.
Oh, okay, but they will go to that saturation, I mean, to that upper bound, I guess.
Yeah, exactly. It will go to this upper bound, but they won't go up.
Yeah, so won't blow up. Okay, good. Good, so the next question is by Xavier Otadzu. What software did you use to implement this network? Is it homemade or an external software like Nest or Brine?
It's totally homemade. I coded it myself in C, to be precise.
In C, okay, wow. So then we have the question by Nicolangelo Ianella. Is there an available preprint of this?
Yes, sure. I don't know if there is a reference somewhere. Yes, the reference it is if you want. It's already published in PLOS, Computer Biology, if you want.
Okay, so not in preprint, but already postprint. Very good. Yeah. Excellent. So maybe there's one more question. Let me check. So here's a question. Basically why the quadratic QIF was used, but in a way you already answered that. But I'll read out the question anyway again, just to see whether maybe not all parts of it were answered. This is Sohail, who wants to know why was QIF used, and in general, what criteria was used to select among the many variations of Hodgkin-Huxley-like models. So I guess the question is, in a sense, why QIF and not the exponential integrated amplifier or any other?
Yeah, I mean, I would say that the advantages of the QIF is, let's say, relatively periodically possible. It's not an Hodgkin-Huxley, but it's in terms of computation, it's not super heavy. I mean, you can run simulation of 20,000 of neurons, or even if, okay, in the result I presented you was with only hundreds of neurons, but we also carried experiments with thousands of neurons, and for sure, with Oxley, it's gonna take a lot more time. So this is, I would say, the main reason to have a trade-off between something quite realistic and something that you can compute in a low time, I think. And also another answer would be that, okay, it was not planned when we made these works, but right now we try to apply this model to robotics, to robots and to embody the framework. And I think for sure with Oxley it would be very complicated. So yeah, this is the main reason, something quite realistic and not too heavy in terms of computation.